Hello biology lovers, my name is Dukunimana Paul. To get a good grade in biology is not still a dream. I'm here to continue the series of the biology topic. Today's topic it's all about food test. So if you are hungry, maybe this video is going to be help you to get food in the future. So you should watch it clearly and you should watch it with full dedication. So we can start our food test. Now the biology, the way I'm going to make it is going to be very fun like this. So just make subscribe so to join my biology family so that you can help me here or here by sharing with others. So stay intact. So biology topic of today, it's all about food test. So biological molecules are the building block of the life. You know, in our body, all of us, we are made of carbohydrates, protein, lipid, nucleic acid such as DNA or RNA, and the vitamins. Not only this, but also minerals and water are the monomers of our body. So the first four organic molecules are called macromolecules. When you say macromolecule, you know, there is macro and micro. Macro, it is when it is large. Means, for example, carbohydrate, protein, lipid, nucleic acid are called macromolecules. While those called micromolecules, they are minerals and water, even vitamins. So the reason why are called macromolecules like carbohydrate, protein, lipid, and nucleic acid is because they are required in large quantity. While those vitamins, minerals, and water, our body require it in a little amount. That's why micromolecules. So in this presentation, we are going to explore how to test for these essential molecules in various contexts. So it means that in this video, I'm going to help you to understand how carbohydrate, proteins, lipids, and even vitamins, specifically vitamin C, how are going to be tested. So stay with me, and this biology should be fun. Whenever you have the disease, you don't 1 week without laughing. So maybe in this video also you can be cured by that disease if you understand me very well. So but I just say it again. If you are hungry, because I'm going to show the picture of the food, please don't serve it, please. So food test. Food number one you're going to test, it is starch. Normally starch, it is one example of carbohydrate. Let's see how to test this starch. So the starch, we're going to discuss in these headings, we're going to discuss procedures. After procedures, you can see the chemical reaction and also the observation, which can show you that this food is containing starch. So procedure number one, you will take 1 gram of starch powder. Means like there is a food you are targeting that there is a starch. You will take 1 gram of that starch powder or that food. You will mix with the water, okay, to make a solution. Then after you will boil while stirring, then you will cool. So after having this solution which is heated and cooled, you will put 2 milligram of the solution in the test tube, okay, and then add 2 drops of iodine solution. So in the summary, you will prepare a solution of this starch containing substance, but you are not sure, you're going to test. Then you will add 2 drops of iodine solution. So what will happen? At this level, let we understand deeply this chemical reaction. It means that the food you are suspecting containing starch, you're going to add the sodium iodine solution. Then the reaction means there is color change which occur due to the chemical reaction between large carbohydrates molecule, which is starch, and iodine solution. So normally this is the test tube where you can take 2 solution in the test tube you are suspecting containing the starch. Then you will add 2 drops of the iodine solution. Now wait for the color change, which is the reaction. So this color change it will be positive result if you get what you call dark blue or blue black color. This suggests that you are having the positive result, means your food is containing starch. So if the starch is the present, the solution will turn from the yellow brown to dark purple, but normally it is dark blue or blue black. This color, it suggests that there is a starch in this food. If the starch is absent, the solution will remain yellow brown. So it's up to you. Pause and review and understand. In summary, to test the starch, you will take starch suspecting to be contained in that food. You will add iodine solution. Then there is color change, which is chemical reaction which take place. Now if there is blue black, that you will conclude that this food contains starch. So thank you so much. This is how we are going to test starch. Apart from the starch, let's continue to the next molecules, which is reducing sugar or non-reducing sugar. But let me talk a little bit here on the carbohydrate like starch are major energy producers in organism. This is simple iodine test allow us to quickly identify a starch in various food samples, from potatoes to processed foods, helping us to understand our diet composition. So means this iodine test, it is helping us to show the food which contain starch. Apart from starch, also there is what we call reducing and non-reducing sugar. So before testing, maybe I can describe what is reducing sugar. Reducing sugar are those sugar which is going to be reduced by Benedict reagent without requiring other preliminary or the other previously treatment. While non-reducing sugar are those sugar which do not directly react with Benedict solution. It means that you should first of all hydrolyze the bond which is making them to be not reacted by Benedict solution. An example for this reducing sugar, you can say glucose, fructose. But when we reach here on non-reducing sugar, the famous example, it is sucrose. So we are going to understand the procedures and even the positive result. So there is the food you are suspecting either to contain reducing sugar. Story number one, you will come and take that food, mix with the water to have a solution. Okay. So means here, let us take example of reducing sugar as glucose. Okay. Means you will take this 1 gram of the glucose powder in the beaker. You will add this 1 cubic centimeter with water. Then you put in the beaker. Means to have a solution. Means pour the prepared solution into the test tube. Okay. Now the next story means the test tube containing a solution which suspected to have a glucose. But we don't test what we know. We are testing whether this solution is containing glucose or reducing sugar simply. Next story, you will add Benedict solution to that test tube. Now, this Benedict solution, you will add 2 ml of Benedict solution to the test tube. Then Benedict solution, normally this solution, they contain copper ions that have a light blue color. It means that this solution in the, maybe here I am supposed to draw. Suppose this is the test tube. So this test tube is containing water, but also glucose. So you will add 2 ml of the Benedict solution. Normally this Benedict solution, they contain copper ion. At this level, because this Benedict solution or this copper ion inside the Benedict solution, they have light blue color. It means normally this solution, they turn to light blue color. So apart from this, we can continue by understanding this. At this level, this Benedict solution now are added to this solution. Now they are light blue color. What the next? The next story you are going to heat means you will take this test tube and then you will heat until you get the observation. So heat the mixture and observe the color changes. Means for reducing sugar like a glucose, as we are talking about the glucose or other kind of reducing sugar, as I told you, the color normally will return from a blue color, okay, because normally if you put the Benedict solution, this solution automatically will become blue. Now the color change will be changing from the blue to light green, then yellow, finally you will get orange, brown, sometimes red. So if you get this a series of the color change, you will confirm there is presence of glucose or there is a presence of reducing sugar. So in summary, how to test, if they ask you how to test reducing sugar, you will come and say you will take the food suspecting to contain reducing sugar or glucose. You are going to make a solution of it, of course, and you will put in the beaker. Next, you're going to add the Benedict solution, which contain normally copper inside. Then your next story, you're going to take this test tube, then you will put on the Bunsen burner, you will heat normally 5 minutes, but you will heat until it seems to be boils. Now the next story, you will wait the series of the color change, where it will turn from blue, light green, then yellow, finally you will get orange, brown. So this is the summary of how to test reducing sugar. So for non-reducing sugar, the story is the same, but the difference is, firstly, for non-reducing sugar, step number 1 is this. Means here it is not glucose. Now for non-reducing sugar, the story it changes. Here is not glucose, is saccharose. Normally saccharose, it is a famous example of the non-reducing sugar. So this saccharose is going to help us on these procedures. Means you will take a food suspecting to contain saccharose, you will put in the beaker and make solution. Then what you will do, you will add Benedict solution as normal way. Then when you heat, there is no color change. Okay? This means that maybe there is non-reducing sugar. Okay. The next story, then what you will do is this. You will just suspect there is non-reducing sugar. Then step number 1, you're going to proceed. First of all, you will add hydrochloric acid to hydrolyze them from non-reducing sugar to reducing sugar. So this acid is the one which will hydrolyze non-reducing sugar to reducing sugar. Then after adding this hydrochloric acid, you will then add sodium hydroxide to do what? To neutralize that acid in the solution. Okay? First of all, you will add acid in that non-reducing sugar. But we don't need this acid. Means after the function of this acid, you will just add sodium hydroxide just to neutralize it, because we don't need this acid. So after this, the next story, you will just now add Benedict reagent. Then after adding the Benedict reagent, you will heat. Then the colors are the same. After adding this Benedict reagent, the color will change from they change from the blue. Now they come light green, then yellow, then finally it is orange brown. The story becomes the same. Means the difference between reducing sugar and non-reducing sugar is because non-reducing sugar, before adding hydrochloric acid, they are not able to react with Benedict solution. But when you put hydrochloric acid, now they hydrolyze it to become reducing sugar. Now you don't need this acid, let you put this base. Now this base is going to kill this acid. Then you will continue your work as the reducing sugar. Benedict reagent, then heat, then observe the color change, which is light green, yellow, finally orange, brown. I hope this is understandable. This test helps to distinguish between different types of sugars in our food. So reducing sugars such as glucose and fructose can donate electron to other molecules. Okay, you are understanding the difference. Means reducing sugar is able to donate electron to other molecules like a Benedict reagent. While non-reducing sugar like sucrose cannot without first being broken down by help of hydrochloric acid. So by XICXI, the story become very easy. I hope reducing sugar and reducing sugar, no one who will say it is complicated, because reducing sugar, take this solution you are suspecting to contain reducing sugar or glucose, then add Benedict reagent, then go and heat. After heating, observe the color change. But for non-reducing sugar, when you add Benedict reagent, when you heat, there is no means it remains blue, no change. Maybe it is non-reducing sugar, so add hydrochloric acid to hydrolyze this bond. Then after, to kill this hydrochloric acid, just use this base to neutralize acid. Then you just add the Benedict reagent, heat, then the color change is the same. Thank you so much. Apart from the starch reducing and non-reducing sugar, let's talk about proteins. I know the proteins are very important macromolecules, so we have a separate unit to talk about each macromolecules in details. So now we are talking about how to test them. So there are many ways to test proteins. The one famous is called the Biuret test. How does it work? Procedures are: you will take this base or this either potassium hydroxide or sodium hydroxide solution. Then you will add to the sample you are suspecting that is containing protein in this amount. Now, shake the mixture. After shaking the mixture, you will add copper sulfate solution. If the protein is present, the solution will turn light blue to purple. So this purple, it is the hint, means this purple, it is a positive result for this sample if it contains the protein. So Biuret test, I'm done with it. Don't wait other notes. Means take 1 ml of base sodium hydroxide, add to the test sample you are suspecting to contain protein. What the next? Shake. After shaking the mixture, then add the ml of copper sulfate, then wait the result. If you get the purple color, that means the protein is there in that food sample. So the common protein source, you know, there are various food sources including egg whites. Albumin means egg whites. There is a milk and also even plant source where you can say soja beans and so on. Not only Biuret test, but there is another test for the protein where they are called the Miron test. Miron test, if there is no such reagent for Biuret test, you can go with Miron's test. Now, how Miron's test says? It says you will take this Miron reagent, it is the chemicals at the market are available. Now you will add to this sample, means the test sample, means the food you are testing. Maybe egg albumin, maybe beans, maybe, means any food you are suspecting to contain the protein. You will add this amount of Miron reagent to this amount of this food. Of course, you make it solution, of course. Now what you will do, you will shake your mixture and then boil, okay? If the proteins are present, Miron reagent change from the colorless to pink. So if this pink color appeared, this is the positive result for the Miron test, of course. Now what you can add on this protein you should know are essential building blocks for the organism, responsible for the structure, function, and regulation. When we say like a structure, we can say collagen. Function, we can say even like enzymes or the antibodies. Regulation, you can say like hormones, and those examples, but we will talk the protein in detail in later on video. This test help to identify the protein content in the foods, which is the crucial for maintaining balanced diet and ensuring adequate nutrition. Suppose that God created us. It didn't tell us this is protein, this is carbohydrate, this is this. So means there is a scientist who just take their time and analyze the food. Now they discover some food, they contain the proteins. So now they use those several ways to test which food they have protein, which do not. So they can use either Burette test or Miron's test. Burette test, the procedures are there, and Miron test, the procedures are here. Pause and review. Apart from the protein, let me talk about lipids. Lipids also, I'm going to talk about the Sudan 3 test, where this Sudan 3 test also are the test for the lipid. Normally, how the procedures are structured. Number one procedure, you will take 5 drops of Sudan 3 solution to this amount of the test sample. Maybe this test sample are olive oil or any kind of the food you are suspecting to contain lipids. So what the next? You will take this you take this reagent, which is Sudan 3 solution, and you add it on the food you are suspecting to contain lipids. Now you will shake, and then you will examine after a few minutes. If the lipids are there, means you will see the lipid layer sitting on the top of water layers with red-orange color. Okay, I hope this lipid layer is going to be observed. Now you will conclude this food is containing the lipid. Normally food test is not complicated. Just take a somehow procedure, then come and see the observation. You are done. There is another test called emulsion test. How does it work? You will take this 5 centimeter cube of ethanol to this food sample test, which is 1 centimeter cubed. So, which is now means after ethanol and added on the food, you will add 5 drops of the water. Then you will shake the mixture. Mixture of how many? Ethanol, food, and even 3 drops of water. When they're using ethanol for the testing lipids, the positive test change from the colorless to milky. When you say milk, it means this food it contains lipids. If you are understanding the concept, just make subscribe, because it is the one way to help this channel. Apart from there is a Sudan test, emulsion test. For the negative result, if the mixture are all water soluble, means if or the mixture are water soluble, means lipids are not present. Because if the lipids are the presence, in this case, the Sudan 3 indicators will form small micellar or drops or droplets and mispersed throughout the solution. It means this lipid layer will not be observed. You will observe a small droplets, which indicate there is no presence of the lipids. Lipids are essential for energy storage. Those are the function of the lipids: energy storage, or the cell membrane structure, hormone production. All of these are the functions of the lipids. Not only this one, these tests help to identify the fats and oil in the solution samples, which is important for the nutrition analysis and the food processing. Understanding the lipid contents help us to make informed dietary choice. As we have seen the previously, means that those food components, either starch, either protein, even reducing sugar or the lipids, all of them are found in our body. Means we should eat the choice dietary, not to eat junk food. Like you are there, you are eating only fried food, or you are eating only sugar because it is somehow sweet. You will die. You should eat the food of the choice just to balance your body, not just to fill your stomach. I hope you are understanding. Now at this level, let me talk about also vitamin C. Apart from starch, apart from reducing sugar, apart from the protein, and also apart from the lipids, let's finally talk about vitamin C, which is our final food component we will discuss in our video. So this vitamin C, it is tested by using what we call DCPIP. Can you repeat? DCPIP. Again, DCPIP. Some people, they confuse on this abbreviation, which means dichlorophenol indophenol. Dichlorophenol indophenol. So those reagents are there to help to identify which food contain vitamin C and which food do not contain vitamin C. So let you have a look on the procedures. So apart from this theory, vitamin C is tested using this PIP, which is dichlorophenols, which has a Bruker anomaly. Like for example, the extract juice from the fruits like orange or tomato. If you want to know if it's containing vitamin C, what will you do? You will add 1 drops of the juice to this centimeter of this PIP solution. So it is better to understand that it is not this PIP solution you will put on this food like orange juice to test if there is vitamin C. No, you will take the first of all test tube. You will put first of all this PIP reagent. Then what you will do? Remember this PIP normally it is blue in color. Okay. Now what will happen? You will take this. You will take this big. You will take this test tube which is containing this PIP, and you will now add drop by drop this juice you are suspecting to contain vitamin C. So for the positive test, what will happen? There is the what you call decolorize this PIP. It means if this solution they contain vitamin C, this PIP which is normally blue in color, it will turn to the colorless. As you are seeing now, it is just you are taking this juice which is orange. Now if this juice contains vitamin C, there is be declaration of this PIP which confirm that there is presence of vitamin C. So like to sum up of our video, to make the summary on this, it is very, very important and mandatory to know this food. I'm just testing which kind of the reagent and what are the positive results. So I just didn't make the table, but I can just summarize it for you because all of them they are there. Now, for example, if it is the starch, okay, this starch you will add what you call iodine solution. Now the positive result will be blue black color, which suggests positive result. Now for reducing sugar, you will add Benedict solution. Automatically they will write green, which will become yellow. Finally you will get orange or red, which is the color which suggests positive result for reducing sugar. So for non-reducing sugar, you don't automatically put Benedict solution. You first of all put hydrochloric acid to hydrolyze non-reducing sugar into reducing sugar. Then automatically you put Benedict solution. Then the color is the same as above for reducing sugar. So apart from these sugars, let you have a look on the proteins. Proteins, we have seen the many ways, 2 ways to test. We have seen the Biuret test, where first of all you will add sodium hydroxide or potassium hydroxide. Then you will shake, followed by adding copper sulfate. Then if there is a protein in that food, you will get purple color. Very good. Apart from this, let you talk about on this Miron test for the protein, which is alternative on the Biuret test. This you will add Miron reagent. This is the reagent. You will add Miron reagent. Then you will get pink color. So this is on the protein. Let you have a look on the lipid. Lipids also you have seen 2, Sudan 3, and even emulsion test. Sudan 3, you will take the food you are suspecting to contain the lipids. Automatically you will add this Sudan black 3, this Sudan 3. Now if there is the presence of the lipid layer on the top, this suggests this food they contain lipid. So apart from this, let me talk about emulsion test. This emulsion test, what is happening? There you are taking your food suspecting to contain a lipid. Then I think you are adding the ethanol. Apart from the ethanol, also you will add the water. Then if the solution turns milky, this is the positive result for the lipid. So apart from this, let me finally on this vitamin C, where you will take this food and drop by drop you will add in this PIP. When you will see declaration of this PIP, this confirms that this food they contain vitamin C. I hope those procedures Go and pause and review. You will understand very well them deeply, and they are very important to understand because it makes us to know this food it contains this food it contains this, and it is a practical examination information. So just understand them deeply. So this test on the vitamin C, it is important in doing this and this. So like application of this food testing, they are very important because some industries on the clinical setting like hospitals or even in the research, these food testing are very crucial to help them to perform their daily duties. So food testing has numerous practical application across different fields like food industry, educational institution like in the lab, in the school. You can't study biology without talking about this kind of food test topic. Apart from the educational purpose, there is also healthcare professionals where in the hospital, for example, there is a disease like diabetes. If you have diabetes, it means you will excrete glucose in your urine. So you can even use this Benedict solution test or reducing sugar test in the urine, then to see if this urine contain glucose. Once you see that observation finally become red or the orange red, you confirm that this urine they contain glucose. Now you can confirm there is diabetes on this patient. So like finally, you should understand this in this topic, understand the scientific behind on this food, those color changes. It means that there is chemical reaction happening in that test tube. Recognize a biological molecule through the chemical test. So practically, you should perform laboratory procedures with accuracy. Here, it means those procedures I have shown to you and even the positive result, memorize them because they are going to help. If you don't know the things, you will not find. Even if you find, you will not know that it is that the one you are searching because you don't know what you are researching. Now, like in industries, they apply testing in the food processing they apply this food test by applying food testing in food processing and packaging, and also personal nutrition. You should eat well, not to fill your stomach. So make informed dietary choices based on the food composition. So like at the end of this presentation, throughout this presentation of the biological molecule testing, you have run it to identify carbohydrate like starch and sugar, either reducing or non-reducing, protein, lipid, and vitamin C using simple chemical test. These skills have applications ranging from laboratory research to food industry and quality control. So for you who reached on this end of this video, just remember to subscribe and share the video for those who are studying biology and even those who want a summary and quick, well explained notes. Thank you. Just study hard, stay happy. I love you.